This is a discussion on the pentose phosphate pathway, otherwise known as the hexose monophosphate shunt. And before I start with that, at this moment, if you're watching my videos in the playlist in order, you should already know there's a lot of things going on with glucose. First and foremost, we know that glucose can be converted to G6P, and then after many more steps can become pyruvate, that is how glycolysis goes. Or if our body needs glucose, usually in the fasted state, we could use that pyruvate to go back to glucose. That is GNG or gluconeogenesis. Also, we could convert some of those glucose molecules to glycogen. So this arrow here represents glycogenesis. Or if we need that glucose, when we have low glucose levels, we could liberate the glucose from glycogen via glycogenolysis. However, another possibility for G6P is to go in this direction, which is literally the first step of the pentose phosphate pathway. So you can kind of think of the PPP or the pentose phosphate pathway as a parallel pathway to glycolysis because just like glycolysis, which happens in the cytosol, the PPP also happens in the cytosol. Though, of course, the products and the purpose of this pathway are very different. In glycolysis, your goal is to produce pyruvate, which is gonna continue on in cell respiration to produce energy, but we don't have something of that sort in the PPP. So, first, in the first step, we have glucose 6-phosphate dehydrogenase, or oftentimes abbreviated as G6PD, converting G6P into phosphogluconolactone. And then after two more steps, we will have the so-called ribulose 5-phosphate or RU5P. These three steps actually constitute the first of the two phases of the PPP called the oxidative phase. It's called like that because, as you know, dehydrogenation literally means oxidation. So you could actually imagine that G6P is oxidized to this product right here. You will also notice that I didn't anymore write the specific enzyme names for the next two steps because they are less important than this. This is actually the rate-limiting enzyme of the entire pentose phosphate pathway. So if you want to inhibit the entire pathway, you could actually just inhibit this enzyme. Although, as you will see later, we don't want to do that because the PPP is important for some essential cellular functions. So, since we do know that this arrow right here represents an oxidation step, literally giving justice to the name oxidative, there should be an accompanying reduction step because oxidation should not happen alone. That reduction is actually seen here, wherein a molecule of NADP or NADP is reduced or hydrogenated to NADPH. Now, this is actually the most important part of the oxidative process because this, again, just like I mentioned, has very distinct purposes, very distinct from the energy generation we have in glycolysis. So allow me to discuss that in detail. First, NADPH serves as an antioxidant, or at least an assistant to another endogenous antioxidant molecule that we have, which is GSH, or glutathione. Antioxidants are molecules that, of course, from the name itself, prevent excess oxidation to happen. Because if a cell actually encounters too much oxidation within its environment, it will be in a state of what we call, or it will experience what we call oxidative stress. It so happens that some biomolecules or organelles cannot tolerate excessive oxidation activity, and some would actually end up being damaged. So, if the oxidative stress is already unbearable for one cell, it may end up actually dying. Of course, that's never a good thing. For example, if the affected cell is an erythrocyte or a red blood cell, it could burst or undergo hemolysis, and of course, you don't want that because literally that's the that's what you don't want to happen. You want cells to survive. So if they die, then what's to discuss after, right? So I'm just gonna use the most commonly mentioned example in references that serves as a contributor to oxidative stress. So allow me to use hydrogen peroxide for this scenario. So let's say that this is the quote-unquote oxidant, which we can think of as the poison. And glutathione in this particular scenario is like the guy which will deal with this poison. Well, come to think of it, since hydrogen peroxide can kill cells by oxidation, maybe to deal with it, you may want to reduce it so that its oxidizing capacity will be gone. So indeed, in order to detoxify an oxidant molecule like this one, you can reduce it so that it becomes a non-toxic product. For the case of hydrogen peroxide, the non-toxic product is actually just water. But of course, we know that reduction cannot happen without a partner oxidation step. Thus, we have to have a molecule which can be oxidized, and that is actually this glutathione. Do note that the moment you write glutathione as GSH, we call that as the reduced glutathione. So now you may ask, how do we call or how do we imagine the oxidized version? Well, you just have to look at this bottom part because GSSG is the oxidized form of glutathione. So how does that make sense? Uh, Remember, or do note, we need two molecules of glutathione for this step. So imagine we have two molecules of GSH. This is the reduced form, right? Well, that's because they have a hydrogen. Remember, if something is hydrogenated, it's also reduced. Okay, those are synonymous words. And if you remove those hydrogens, the hydrogenation means oxidation, right? But in order for this to be complete, actually, the moment that you take away these hydrogens, these two sulfur atoms end up binding to each other. So, once they are already oxidized, they are actually joined together. Thus, 2 of GSH, the one with the H, is the reduced form and is the active form, as you can see here. And after oxidation by glutathione peroxidase, the H's will be gone, we get GSSG. Of course, we know that GSSG is not anymore gonna be helpful in the detoxification process. That is why it is important to note that we could recycle this oxidized version back into the reduced state via glutathione reductase. Of course, the reductase will reduce our glutathione here to go back to the top so that this one is regenerated and we could further perform detoxification. However, we have to again go back to the idea that reduction cannot happen alone and must have a partner that is oxidized. And that is now our NADPH. We could imagine as if NADPH is willing to give up their hydrogen atoms so that when these hydrogens are removed, you give them back to the GS molecule. Not really back, but you give them to the GS molecules. The H returns, the disulfide bond breaks, and then we're back to the reduced state. So like we can think of like, oh GS, you have no H, I'm gonna give you H. So now please do your work again. So just like I said, you can think of NADPH as an assistant to our glutathione. And what do we use to make NADPH? This oxidative phase of the pentose phosphate pathway. That's why you have to understand that I did say a while ago, you don't want to inhibit this enzyme because as much as there are many poisons or toxins in our environment that cause oxidative stress, it is important that we have a constant amount of NADPH to fend against these poisonous substances. Otherwise, our cells will die. So, you also have to know that there are actually some people who are born with a genetic deficiency of this enzyme. You call these people as G6PD deficient, and it's actually one of the most common enzyme deficiencies in man. So, what happens if I have G6PD deficiency? Well, of course, since I have lack of this enzyme, I have lack of NADPH. If I have lack of NADPH, then I would have the lack of the active glutathione. Therefore, the detoxification process would be slower than usual. These molecules could accumulate, and therefore it is more possible for the oxidative stress to cause cell death or hemolysis. That is why one of the manifestations of G6PD deficiency is the relatively easy bursting of the red blood cells of these people. So you can kind of say that their erythrocytes are fragile. Thus, compared to a normal person, a guy with G6PD deficiency would have an easier time bursting or popping off their erythrocytes, which is not actually a severe scenario. It doesn't happen spontaneously. That's why there are so-called triggers that would have that a person would have to encounter before the red blood cells break. If the triggers, like some food or some drugs, are not encountered, luckily, that person is actually very much normal. Okay, but still, the sheer popularity and frequency of this is something worthy of discussion. Now, after the antioxidant use of NADPH, another use of NADPH is biosynthesis. What I'm trying to say here is that NADPH can serve as a supplier of hydrogen for hydrogenation or reduction processes, because you may remember, if you've watched my intro to metabolism, that many anabolic, or in general, the pattern for anabolic or building up processes is they are reductive processes. And of course, if you want to perform reduction or hydrogenation, you need to have hydrogen sources. It so happens that most of the time, the hydrogen source we use is NADPH. Thus, we would actually encounter NADPH, for example, in the synthesis of lipids like fatty acids and cholesterol, just because of the sheer nature of anabolic pathways. Yeah, so that's NADPH. And if you're just watching this video for the oxidative phase, you could actually stop here. But if you need to know also about the non-oxidative phase, carry on. The non-oxidative phase is actually very different because since it's not anymore oxidative, we're not producing NADPH, so it's an entirely different story. It so happens that we pick off from where we left off a while ago, which is RU5P. RU5P is a pentose, okay, which could isomerize into two other pentoses, namely ribose 5-phosphate and cellulose 5-phosphate. This should reveal to you that ribulose and ribose are not the same. If you remember our discussion on carbohydrates, if you watch that, usually the fact that there's a UL in the middle of the name means that you have a ketose molecule, right? So ribulose is actually the ketone version, and ribose is the aldehyde version, okay? And the point here is that in order for us to proceed discussing the rest of these things at the bottom, we can imagine at this point that we originally have two molecules of RU5P. One of them has become R5P. One of them has become XU5P. Now, if we think of it that way, we now have two pentoses with us. So if you try to sum their carbons, that's 5 plus 5, we currently have like 10 carbons at our disposal. Now, what I want to imagine is like this and this are two separate molecules of clay, and then you try to merge them together and then, play with it, you roll it, and then you mix it together, and then you try to chop it off again into two products, something like that. Wherein, of course, we need to follow the law of conservation of matter. In this case, if we start with 10 carbons, then every time we mix and then split these, we should still have 10 carbons. So for example, after transketolase acts on these two sugars, your carbon count should still be 10. So notice our products. One of them is heptulose 7-phosphate, or S7P. I'm not actually sure if this is S7 or SU7P. Okay, but the name is this one, and it's a 7-carbon sugar. It's a heptose. And then the other one is GAP. This one is glyceraldehyde 3-phosphate, and I'm abbreviating this because this is part of glycolysis, something we've already discussed already. If you are watching my videos in their proper order, so this contains 3 carbons. 7 plus 3 is 10. You could once again mix them together and split them into 2 using a transaldolase, and you get 2 unique sugars once again. One of them is erythrose 4-phosphate, which is a tetrose, and then the other one is fructose 6-phosphate, of course, a hexose. So once again, 4 plus 6 is 10. And really, the way that you tackle the non-oxidative phase really depends on what you want to study. Because if you're studying, for example, human biochemistry, we will actually be focusing more on this, which I will emphasize in a bit later. If you're more into plant chemistry and you're just watching this video as a review, you may be dealing with E4P more because there are some pathways for the synthesis of some secondary metabolites to start with this. So it really depends. Or maybe if you're just trying to connect this with glycolysis, these two would come out as the most significant for you. But just to make the situation more complete, based on how many how some references would depict this in their figures, let's just imagine that I have an extra cellulose 5-phosphate with it. Remember, at this point, our end products so far are E4P and F6P. Now, imagine we add an extra cellulose 5-phosphate, which will interact with our E4P using a transketolase. So the transketolase mixes these two and then yields two products. Apparently, one of them is F6P. And in that case, that this F6P is different from the original one I boxed a while ago. So you can think of it as having two F6Ps at the moment. And then the other one is gap. Actually, I think this is not anymore correct. This is just one gap. The first gap is already gone because it has been converted to F6P. So this is not a second gap. Okay? This is just one gap. And if you try to think of the situation as you originally start with uh, in this arrow, for example, if you start with 4 plus 5 carbons, if you start with 9 carbons, what are your products? 6 plus 3, which is also 9. You've conserved again the number of carbons. Or maybe if you think of this in the larger picture or in the bigger picture, like you start with 5, 5, and then you add an extra 5 carbons. So 5 plus 5 plus 5 is 15. How many carbons do your products have? Remember, the overall products we have so far are 2 molecules of fructose, so 6 times 2 is 12, plus 3, that's also 15. So, uh, as you can see here, depending on the perspective you are trying to employ, we're still being consistent with the total number of carbons. Nothing is lost, nothing is gained. They're just shifted around and played around with. But what's the point of this, you may ask? For some plants, I did mention you may focus here, or some cases you may focus here. But if there is something quite universal to all organisms, why we perform the non-oxidative phase, I think it will all boil down to this molecule, or this sugar, ribose 5-phosphate. That is, I clicked the wrong thing. That is, regardless of where you start, okay, you could actually end up with ribose 5-phosphate. So now, you may ask, why do we need the ribose 5-phosphate and why is it universal? I want you to pause, or maybe if you want to hear the answer, then just wait for it. Where did we ever discuss a situation having ribose and phosphate at the same time? And maybe you may remember that ribose is our sugar for nucleic acids, right? And nucleic acids are bound to have phosphate because that is what defines a nucleotide. It's as if I'm saying here that there's only one thing missing here, right? Like all we need is a base like a purine or a pyrimidine, and we now have a complete nucleotide. I'm saying here that we need this because this is actually two-thirds of a single nucleotide. And remember, nucleotides are part of DNA and RNA. All organisms must have that. That's why I'm saying that, among other things, one thing that is universal for all organisms, be it human, bacteria, plants, animals, other fungi, in the non-oxidative phase is the synthesis of this because this is literally where we get these parts of our nucleotides. For without them, how are we gonna make our DNA and RNA, right? So as you can see here, the purposes of the oxidative and the non-oxidative phase are very distinct. For the oxidative, you have to focus your eyes on this molecule, and then for the non-oxidative, you focus on this, which, as I have explained, are very, very different in terms of what they do. As a final note, I would like to explain why the PPP is sometimes called the hexose monophosphate shunt. You may ask, how are these two very, very different terms referring to the same pathway? And the answer to that is perspective. You can call it the hexose monophosphate shunt if you focus your eyes on the starting material, because remember, everything started with a hexose with one phosphate. That's why you call this a hexose monophosphate shunt. And this is a shunt because you can treat this pathway as another extra pathway, wherein the other pathways include glycolysis and glycogen metab, right? That gives justice to this term. And obviously, the reason why we call this the pentose phosphate pathway, because as I have mentioned, our end product for nucleotide synthesis is ribose 5-phosphate, which is a pentose phosphate. One last note, since we did start with a 6-carbon compound and end with a 5-carbon compound, you may ask, where did that 1 carbon go? And just to complete the story, that actually was lost in the third step of the oxidative phase as carbon dioxide. Okay, so just to make sure we don't have like plot holes here. So that's all I want to share with the pentose phosphate pathway.